Hello, YouTubers and students. Uh, I'm Dave Soriano. I'm a chemistry professor with the University of Pittsburgh's Bradford campus in Western Pennsylvania, USA. Uh, I don't like to do carefully rehearsed videos. I like to do things spontaneously. I hope you will understand that when I show you the contents of this video. Um, I am going to, in this very simple introduction, show you how to approach the Protein Databank. That's online. You can access it with Google or browser, Marilla, what have you. The Protein Databank, and uh, I've been using this for years in modeling studies. And what I usually expect is that my students who enroll in an advanced biochemistry course with me, I'm an organic chemist actually, but I teach undergrad biochem. In the second semester, advanced biochem, I will show them how to begin to do molecular modeling, uh, small organic, small molecules. They may or may not be candidate drugs, or they may actually be prescribed medications, and receptor sites, proteins, be they embedded in a membrane or an enzyme. What I want to show you here is a very timely application, the coronavirus spike protein. And that is, as probably everyone is aware now, the projectiles, I'll call them, the proteinaceous projectiles on the surface of the corona. And it is an integral part of the docking phenomenon. For example, it would approach the virus, would approach the ACE2, and that would be the angiotensin converting enzyme 2. Number one is really a hormone, ACE1 is really a hormone, but ACE2, and this is where the virus docks via the surface of the spike protein to the ACE2 receptor site. The membranes blend, and then the RNA genome of the virus penetrates the host cell and takes over. What we're seeing here, and I never believed for once that the Chinese scientists, I don't want to get into any of the politics, but I don't believe they were doing anything to create bioterroristic viruses. I think they were simply studying them to try to come up with vaccines. Uh, but let's leave that to the side. If they were doing that, they wouldn't publish their work. So what we see here, a catalog number in the Protein Databank is 5GYQ, and that's the putative receptor binding domain of the bat-derived coronavirus spike protein. And there are a few other X-ray images that have been uploaded to the Protein Databank. If you go to the home page of the Protein Databank, they will direct you to all of the corona images, 3D images that have been uploaded. Now, for undergrads, we would download the file as a .pdb. So it would have the X, Y, and Z, the three-dimensional coordinates, and that's what you would then save. Now, you can do some very good modeling here online, but let's go directly to what I want to show you. And what I will do now is X this out, 5gycube.pdb, and then we will go to Scigress. So I will bring that up, and it will be with us in a moment. There we are. And I've already opened it up, Scigress, and I'm going to go to File, New Project, let me call it, and I'll just click on here File April, let's say, and I'll save that. Um, I'll open that up. Um, and we'll put that as new object within molecule. Okay. And we are keeping this in April 5. You can see at the top, I'm using a Mac, chemical sample 6. So what you have are open projects, and within a project, a new object, a new study. So what I'm going to do now within this project previously established, I'm going to import that file. So now I'm going to go to PDB, and incidentally, you can go to MolView, MOL View online. It's in the public domain, and you can draw out 2D sketches of drugs, create 3D images, and save them as .mol. And Scigress platform, which I use here, will read that. Let me just show you in passing. These are the different files. Scigress, but there's the mo.l. So you can then bring that right in. I don't like drawing 2D sketches. Well, you can't in Scigress. You have to draw in 3D from scratch. That's not good for beginning students. And even for those that excel in organic, they've had limited experience in this type of work. So it's always best to start with a 2D sketch, then bring it to a 3D, make a few cursory views of that, and then go from there. So anyway, let's go back to this program. We'll go to all files. And there's the one I was talking about. We'll open that up. This is just an initial view. Now, you're not going to do any modeling with the ball and stick model. I mean, this is not some small organic molecule. We're talking about, in this case, it's only the portion of the spike protein of the virus, only the portion that's involved in docking to the host cell. Now, what we'll do with actions, and we'll just go through this very slowly. I'm going to suppress the hydrogens. They have been suppressed. And then what I'm going to do is say, well, how can we go along from here? Well, what we can do is color by residue. Now, look at this. This is a very good application in Scigress, and it's a very good program, for professional researchers use it in doing modeling of candidate drugs before going to the lab where it's time and money. We're going to portray any in this portion secondary structure. Primary structure, of course, is the sequence of amino acids. And here we're going to look at this portion of the stem protein, and we're going to portray the helices. That's one of the 2 or 3 common motifs in proteins. Helix is yellow, beta sheet red. And we'll go with that. And then we can start to see we're making some progress. And then what we can do is backbone, and we can do ribbon or trace. And I'll go to single thread. And now we're making progress. Now, most life scientists I would take do not begin to do modeling work in this fashion. They would show the common arrows for beta sheets. Alpha helices, of course, are corkscrew. This is a way that I, for example, like to study this. And the dots are water molecules. So there you can see a helix. This portion of the spike protein really has more of the sheets, and they're parallel sheets. And there's a single, up at the top in the docking area, a single alpha helix. And then, of course, where it docks is the course of study. In a subsequent video, I will show you exactly how we're going to keep the very critical number of residues. I am, at this point, aware there are 4 critical residues in the docking phenomenon. Now, don't hold me to that. But what we will do is, from the scientific literature, ascertain what we can of the ones that are critical right at the front. If this is a war with the virus, then you're looking at the front here. And we must keep in mind that many people have suffered, died, economic loss, and we're looking at the eye of the hurricane here. So what I will do, let's see if I want to show you anything else right now. Protein, we can do the sequence. And there, PDB, the protein database, will show you that too. But then you get the sequence of all the amino acids. Now, I'm old school, and I know the three-letter symbols for the amino acids, but most modern molecular biologists and molecular scientists in life work, life science research, probably are using the one-letter symbols now. And I always have to have a little conversion factor so I can tell that R, for example, represents the amino acid arginine. And I do think there are two critical arginine residues that seem to be involved in the docking phenomenon. Now, what I'm going to model is to get right to the forefront, and I want to do what's called in silico site-directed mutagenesis. I want to be able to replace one of those residues with another and try to get a feel on how that would increase or decrease the binding to the ACE2 receptor site locale. And site-directed mutagenesis in the laboratory was extremely expensive a number of years ago. It's getting cheaper. And I want to see if this virus could actually bind better in modeling studies with some other amino acids at the forefront versus the ones that are there right now. And the bottom line is I want my undergraduate students to leave our campus knowing the rudimentary aspects of how to do molecular modeling. So if they go on to graduate study, they would have the tools needed to be then guided by professional research scientists, graduate professors, or to go to industry, let's say, and work in a biotech company, and at least say I'm at the point that I can, carry out whatever modeling studies you want me to do. So this is great stuff for the young people. Okay, so that's all I'm going to show you right now. And I will be uploading other such videos. And look for the other ones I've uploaded already. For example, how to take a 3D sketch from MoleView and import it right into Scigress. Okay, thanks for watching. Bye for now. If you have any questions, just send me an email. Take care.